The title of my talk is Designing Cellular Information Units. And we're in our lab, our research is generally driven by the understanding and developing strategies to manage and treat tissue injuries. If you look at tissue injuries across different circumstances and also different organs, you'll find common denominators that includes, for example, when injury is acute, you'll see inflammatory response and barrier injuries. A lot of times when injuries are repeated on the same organ, which is a chronic injury, these conditions still remain. But also there are conditions where it become more permanent, including degeneration and formation of fibrotic scar, which compromise the organ function and leading to death. The reality is that our body does have ability, or equipped at least, to regenerate upon injury, and that's called stem cells. Uh, Many organs have adult stem cells, but the reality is that they're also surrounded by and constantly receive signals coming from their surroundings, including secreted factors, other cells, as well as more of physical factors such as extracellular matrix and the shear force. And what happens upon when tissue is injured is that these signals coming from the surroundings, which are called either microenvironment or micro niches, they themselves become compromised so that they can influence stem cells in a negative way, including cellular communication, exhaustion, and senescence. So our research we've been looking at ways to rebuild regenerative capacity of the body across different scales. And today's focus is going to be that the key concept where we can build microscale environments or micro niches as programmable units so that we can not only direct cellular behavior, but also do so in a predictable way. And briefly, we also wanted to look into ways to achieve more of a global impact to regenerate the tissues, injured tissues, over distances globally, systemically, by looking at how our body makes these kind of nanoscale mediators that can travel across the body and influence their behaviors. But that's unfortunately not something that I'll be talking about today. So for this part of the talk, the micro niches, there are generally two parts where the goal is to discuss how microscale environments can guide cellular behavior, but also remodel the environments that are already compromised beyond the immediate cellular environments. If you think about how we can go about designing micro environments around cells, it'll be very useful to look at how our cells actually do it, because in the end of the day, the extracellular matrix or the micro niches are encoded. A lot of times they're encoded by in the genome. So cells lay down and create the extracellular matrices. And one interesting example is to look at chondrocytes, where you actually see these cells and they form these dense matrices immediately around them. They're called pericellular matrices. But this also is generalizable across different types of cells, as you can see in the smooth muscle cells, as visualized by just essentially the beads. And the parts that are not penetrated form these kind of extracellular matrix layers. In fact, some studies show that these extracellular matrix layers around immediately locally deposited around cells are highly elaborate, consisting of hyaluronic acid, proteoglycan that can be sensed by receptors like CD44. So how can we go about engineering these kind of a local microscale environments around cells, preferably around single cells? Um, so we've been using this biomaterial called alginate, where they've been used in across different scales, length scale, as a hydrogels, as a tunable materials. And the advantage of using hydrogel, alginate hydrogel, is that they can be they can undergo gelation in the presence of divalent cations, so really rapidly and also in a high affinity. Um, they're carboxylic, they're acid, so they're a bunch of carboxylic group, which means that relatively simple chemical mechanism, like a carbodiamide chemistry or NHS chemistry, can be used to introduce any type of amine containing groups, such as if you want to functionalize adhesion peptides to these hydrogels to induce the adhesion of cells. Um, Alginate hydrogels are pretty amenable when it comes to tuning mechanical properties, such as elasticity as well as viscosity. And cells can be either seated or encapsulated across different scales. So in order to create literally the micro scale environment around cells, single cells, we've taken advantage of droplet microfluidics, but also in order to do so in a more deterministic manner, at least in terms of the final product, while allowing us to tune the properties around the cells, the gel properties around cells. We leveraged these precursors, which is a calcium carbonate, that are and absorb them onto cells, and we mix them with alginate so that these precursors alone wouldn't undergo gelation, at least immediately. It'll take many hours to undergo gelation. In the oil phase, when droplet is formed, oil contains the acetic acid, so that the acetic acid releases calcium ions on the cell surface, which results in rapid gelation and inside out gelation of the alginate gel around individual cells. And what's important here is that what's interesting about this approach is that because the gelation would occur only when there is precursors, and precursors are only present around cells. This results in the dramatic substantial reduction of the empty microgels. But the product that we can obtain are the ones that contain individual cells coated with the hydrogels. Why is this interesting from more of a physical perspective? If you do a simple calculation where let's say cell diameter radius is 5 microns, typical radius, and the thickness of the extracellular matrix, and you do the volumetric calculation, assuming that everything is a sphere, then you find that most of times, even when, let's say, the matrix is like 10 microns in thickness, the volume is predominantly dominated by the matrix. But when you go below 10 microns, this is when you actually see the meaningful variation in terms of the cell versus matrix ratio. So the ability to control the amount of matrix around individual cells at this scale allows us to really study the cellular behavior and their impact in this case. So in order to vary the amount of hydrogels around individual cells, we basically varied the device flow parameter as well as the cross-linking. And we demonstrated that it is possible to vary the amount of the hydrogels around individual cells, ranging from 1 micron in thickness all the way to hundreds of microns in thickness, while maintaining the intrinsic material properties such as the elasticity as well as the viscosity. And this work is done by a former postdoc of the lab, who's now a faculty at Colorado State, Seung-Wan Wong. And One thing that we did is just to look at cellular behavior, how cells shape, undergo shape changes over time by encapsulating individual bulk gels. And with the softer, relatively softer gel with a certain adhesion ligand density. And we use mesenchymal stromal cells as a model cells. And when we encapsulated cells, individual cells in without the presence of adhesion ligand, no matter what thickness that you encapsulate, the cytoplasmic volume remains constant, similar, right after encapsulation, around 6 hours after encapsulation. However, what's interesting is that when you start introducing RGD peptide into this adhesion peptide, we saw that there is a rapid isotropic expansion of the cytoplasmic volume when they're encapsulated in thin RGD peptide gel compared to thicker and bulk hydrogel. What's interesting is that this also translates to the expansion of nucleus volume, where you also see the increase in nucleus volume in the thin RGD gel. I wouldn't get into like mechanistic detail, but we figured out basically that there is a combination of ion channels that play important role in regulating this process. But I wanted to highlight what the consequence of this phenomenon is. So as a result of cells being able to undergo isotropic expansion rapidly, but also in a sustained manner over time, we measured it over 3 days and this expansion still occurs. We basically see that there is an increased membrane tension around individual cells when they're in the thin hydrogel compared to thicker microniches, as measured by the flipper dye, which changes conformation. And this changes in conformation upon high tension results in a slower decay in fluorescent lifetime imaging, fluorescence lifetime. So that way we'll be able to measure or understand the membrane tension of the live cells that are encapsulated in the microniches. Another important consequence is that we figured out the downstream pathways that ultimately results in the matrix production. To understand whether the cells are able to sense the microenvironment and start making the new matrices, we used the metabolic labeling where we fed cells with the azido version of methionine, or it's called azido homoalanine. And then we did an encapsulation in thin versus thicker hydrogels and then culture them. And then we used a decodite to detect the nascently produced proteins. And we show that the amount of newly synthesized proteins deposited around single cells is increased when cells are encapsulated in thinner gels, the microniches compared to thicker microniches. And there is this relationship there. So this suggests that by simply encapsulating cells in thin microniche, it is possible to direct their cellular behavior, not only in terms of volumetric expansion, but also synthesis of the new matrix. So in other words, cells are able to sense how much matrix they're already surrounded by, and then make new one if they're needed. Another thing that I wanted to highlight here is that the reality is that the matrix around cells will be heterogeneous. So to model this phenomenon, we took advantage of the fact that the microfluidic, in the micro scale, the fluid flow tends to be laminar. So by using the 3 different channels and progressively reducing the flow rate in the middle while increasing the flow rate in 2 side channels, and combining with this chemical approach, we managed to encapsulate individual cells in 2 different alginate compartments where each alginate is independently defined in terms of their biochemical components, such as adhesion molecules. And by doing that, we were able to see control how cells, the polarity of the cellular adhesion around the matrices. So for example, we by using flame approach, we show that the alginate that contains the RGD on one side, cells are able to interact with it compared to the ones that that is missing. So by using again this membrane tension reporter, we show that the presence of RGD adhesion ligand and only on the one side results in the higher membrane tension compared to the one without RGD. And this, we also vary the different concentration as well. And this actually results in an interesting phenomenon where the cell volume expands only towards one side compared to the other, resulting in the elongated shape of the cell. So far, by leveraging droplet microfluidics at the very small scale, to the level that cells are able to sense and understand them, we're not only able to achieve isotropic expansion in new matrix synthesis by decreasing the hydrogel amount, but also by decreasing the symmetry of the adhesion ligands, we managed to achieve elongation and the polarization of the cell. So thereby demonstrating that the micro niche design sculpts and stabilizes single cell behavior. We wanted to also, but so far, all of these studies were done by changing parameters one at a time and looking at somewhat collective behavior. So we wanted to use the power of picoliter scale droplet microfluidic approach to simultaneously generate the micro niches with different sizes and encapsulate them, and leveraging unsupervised clustering approach to study the morphological parameters and how they change over time. And this result basically shows that in the thicker microgels, cells actually undergo two different kind of a trajectory where one trajectory they remain unexpanded, while other trajectory they actually develop actin cytoskeletal formation, while cells in the thinner gel consistently undergo expansion in volume. We can also take these parameters to predict what kind of environment that cells are present 80%, 82% of the time, which is higher than just by chance. And we're able to combine this information to better understand the cellular heterogeneity over time. So this is something that we plan to use moving forward. Just to quickly wrap up the second part of the talk, we wanted to understand the impact of controlling the cellular individual cell behavior by using microscale environment, by looking at how this environment results in remodeling of the fibrotic scar. And just to quickly let you know, I mentioned fibrosis. It is a mechanically driven failure of the tissue function. For example, if you take lungs, it's a healthy environment. There's a gas exchange, but upon fibrotic injury, there's a scar formation. And when scar is not resolved over time because of the chronic injury, the scar formation interferes with the gas exchange, ultimately results in breathing difficulty and death. The fibrosis in terms of mechanical property is actually seen across different tissues, which is characterized by a significant increase in tissue microelasticity. An important cell in this context is MSCs, mesenchymal stromal cells, which not only have ability to undergo differentiation, but also they act as a parasite to secrete these paracrine factors that can influence different kind of other cell behaviors of other cells, including immune cells and vessels and etc. However, in fibrosis, these functions become compromised. So it doesn't although MSCs have this ability to maintain tissue homeostasis, providing directly injecting them into the scar wouldn't really help because the scar itself can compromise this MSC function. So we did basically the thought experiment where what if we can supply the body with MSCs, but also protect them in the microscale niches in order to allow them to secrete these therapeutically relevant molecules as a means to resolve fibrosis. So the advantage of using, again, the single-cell encapsulation here is that we can not only deliver the cells with a defined microenvironment to deep tissues like lung airways, but also we can continuously present these cells with locally defined signals that can then enable them to restore host environment by secreting therapeutic paracrine factors. And then so the first generation, we indeed demonstrated that by using this single-cell encapsulation, but embedded with the inflammatory cytokine to locally challenge MSCs in this environment. We managed to achieve this property where the collagen, the enzymes that degrade collagen is constantly secreted. This is just gene expression studies. And we show that the collagen degradation takes place even when cells remain within this environment in a manner that is dependent on the enzyme against collagen called MMP. So this shows that cells secrete paracrine factors to degrade collagen when they are in the right type of environment, microniches. But the issue with this approach is that they can be cleared rapidly because the hydrogel material are easily opsonized. So we developed recently the second generation therapy where we tested different types of molecules to reduce opsonization and to evade the clearance by macrophages. And we initially tested the zwitter ion molecules to reduce the absorption of proteins. And indeed, the residence time of cells is encapsulated cells is improved by about threefold. But we wanted to do better than that, and we introduced the peptide coming from CD47, which is the natural proteins that are expressed by pretty much all types of cells. They act as a marker of self, and so they limit the phagocytosis process by activating their counter receptor called SIRF-alpha. And grafting this peptide onto the gel actually resulted in substantial improvement in the residence time. So, and this proves to be useful in an animal model where scar formation is more permanent. And we managed to do that by using the repetitive bleomycin model by delivering every 2 weeks or so, 3 times. And after that, after about 4 weeks, after this experiment, when the scar formation takes place, we then deliver this treatment and did analysis. And across all board, including the collagen deposition, as well as epithelial restoration and ultimately lung function, we show that the gel-coated MSCs that grafted with CD47 peptide, but not the ones that are not grafted with this peptide, resulted in the resolution of the more persistent fibrotic injury. So basically, just to wrap up my talk here, there is a huge potential in combining biomaterial design with microfluidic droplet microfluidics, and combine this with the mechanobiological studies to guide individual cell behaviors, as well as to remodel the host environment and also delay the clearance. But ultimately, the incorporation of synthetic biology and advanced materials down the road will hopefully enable us to precisely control these behaviors that could be broadly useful for treating the therapeutic conditions, treating conditions like chronic injuries. So with that, I'd like to acknowledge my former team, former institution. I moved to University of Michigan recently with some members, collaborators, including former and new, as well as funding sources. Thank you.